Primary diagnosis is when a pathologist looks at a slide and through really kind of a miraculous process, the visual information goes in, something happens in the brain, and then, either instantaneously or a few minutes later, or maybe after some tests, some words come out. The words get into the report and dictates like everything to follow with the patient. So what's in the black box of the brain that's going on? So one is something called features. Features are what you learn in histology class. Here's a nucleus, here's a cytoplasm, here's a fat cell, here's an infectious agent. The second one is context. So you can probably think of some examples where if I show you some features in this one history, but then I show you the same features in a different history, your primary diagnosis might be different, right? So it turns out the context can be really, really important. And then the third one is language. So think about how important that we all have shared meanings of a set of words that come out of the end, right? Invasive versus in situ, pleomorphic versus anaplastic. Like these automatically, whether you like it or not, I say these words and the part of your brain that makes pictures is already flashing pictures in your head. We don't really understand what's going on. All we have is really the diagnoses to compare.
Learn about the newest digital pathology trends in science and industry, meet the most interesting people in the niche, and gain insights relevant to your own projects. Here is where pathology meets computer science. You are listening to the Digital Pathology Podcast with your host, Dr. Alexandra Zhurov.
Welcome, my digital pathology trailblazers. Today, our podcast guest is Dr. Keith Wharton from Roche Diagnostics Solutions. And our topic is digital pathology validation studies to get a whole slide imaging system cleared by the FDA. And Roche received clearances for their whole slide imaging system that includes two scanners, the DP200, and that was in the summer 2024, and DP600, and that was at the end of 2024. And we will be talking about what actually needs to be done and why to achieve this milestone, and how this compares throughout the industry, not just what Roche did, but there is a paper in the making that we're going to be talking about. So good to have you on the podcast, Keith. Quick background of how we know each other. We actually worked together previously, and we co-authored a paper. Title of our paper was Tissue Multiplex Analyte Detection in Anatomic Pathology, Pathways to Clinical Implementation. So we brainstormed and geeked out on how multiplexing can be used in the clinic. But we always start with the guest first. So, Keith, let us know a little bit about yourself, your background, and what is your role now at Roche Diagnostics?
Thanks, Alex. It's so great to be here. And I just want to say thank you to you for all the advocacy of digital pathology that you have accomplished in the last few years since actually we used to work together. So that paper was published in Frontiers Journal in 2021, and it explores
that
a
was
topic
when I
that
incorporated the podcast and blog, Keith.
Yeah, I figured you were just thinking about it. I think my motivation for that paper, and if you remember, it was just this widening gap between what's technically possible, all of the discoveries we're making with digital pathology, spatial multi-omic biology. We'll talk about that a little later, and what fraction of that is going to impact patients in the near future. I think it's a widening gap, and that's one of the reasons I'm very passionate about what we do at Roche. And I think today's topic is very relevant. And for that reason, we're going to touch on some topics like the pixel pathway. That paper's not obsolete yet, so even though it's 4 years old. So my background, I started out as a chemical engineer, became a medical doctor, and then trained to be a pathologist. But I did the whole research spectrum from stem to stern. As well. I did a PhD in molecular biology, a postdoc. I was a faculty, and then I was actually associate dean for medical education for a little while before entering industry in 2009. So that was 16 years ago. And I've been really lucky to spend about half the time in pharma and half the time in kind of the diagnostics, life sciences tool space. And so I've been then with Roche Diagnostics since 2022. So it was after we worked together. I'm in a global medical affairs role, and I mean, you could guess what I'm covering, right? I'm covering digital pathology and multiplexing. So if the shoe fits, wear it, and a handful of other things. Roche is a very rich company from a scientific and product development standpoint. So I'm really thrilled to be here at this point. And I will say I've been really lucky from the standpoint of background as a pathologist in industry to actually do pathology the whole time. A lot of MDs go into what I'd call clinical development, medical affairs, or drug safety. These are all great things to do, and they're really important, but none of them are actually pathology when you get right down to it. And you know this as a veterinary pathologist. And then actually in contrast to DVMs who go into industry, who mostly do toxicologic pathology
Yeah,
of
we
one sort
actually
or
do
another.
pathology in the
Yeah,
industry, yes.
I was going to say, you guys have a career path. I found like MDs really don't have this career path. So I had to
That is
kind of
true. I
piece it
never
together.
looked at it like that.
Yeah, I had to piece it together, jump around. In the pharma roles I was in, I was the only MD in departments of all DVMs. So from the standpoint as an experimentalist, I loved it. So I was like, I'd call myself the token MD in that group. Because as you know, a lot of those folks do combined safety and efficacy studies. So it's actually really important to know what you're looking at, and all about the target, the modality, and try to interpret the lesions in context of potential safety signals or efficacy. And so I guess after having pieced that together now for the past 16 years, I'm kind of happy. I've been able to survive in industry as a MD pathologist. And I'm also proud, I've been able to publish like 12 papers and a book on, that's very relevant to the topics that we're going to talk about. So within Roche, Roche Diagnostic Solutions, most of what I support would be what we call the legacy Ventana. Ventana started as Ventana Medical Systems, actually founded in 1985, I believe, by Tom Grogan, who's a pathologist. And I don't know if you're familiar with him. He was actually one of my professors in medical school. So I had the privilege of meeting him as a medical student. I think it was 1987. I was a young person trying to figure out what I wanted to study and do. And actually what he was actually doing didn't excite me, but that's okay. He was actually very, very encouraging. And that kind of has set the tone, the type of individual he is actually to this day. Within Roche Ventana. So he retired full-time from leadership, I think in 2017, but he's still around and he's still really inspiring to people. And I guess the last thing I should mention about Tom is that actually right before the pandemic, he wrote an autobiography that is not only actually a really interesting autobiography. It's called Chasing the Invisible.
I need to review it on the podcast.
Yeah, he would love to talk to you, actually.
We're going to link to this book in the description.
So 2019, I think, Chasing the Invisible. So part of it is his kind of odyssey through founding Ventana and all the ups and downs he went through. He's really revealing like how stressful it is to take a company from, twinkle in your eye to acquisition by Roche, in this case, in 2008. So, but the other piece of that you don't realize is what a major role Ventana has played in the advancement of precision diagnostics, really through automated staining, companion diagnostics, and now we're entering this era of digital pathology and entering the next stage. So, I recommend you look
you
up
need
with Tom
to introduce me. Maybe you can do an
we
introduction.
can do that.
And then...
We can do that. Chasing the Invisible. Of course, he called it that because when you look at an H&E, like you can't actually see the stain biomarkers, but he is a hematopathologist, was doing a lot of, in the initially manual immunohistochemistry, and that was like the inspiration and motivation to actually roboticize the whole.
To automate.
Yeah.
Oh my goodness.
And he wasn't the only one, but he was certainly one of the leaders. And as you know, it's one of the leading platforms today. That's in pathology labs. Yeah.
I need to read the book because what's happening in somebody's mind to design something like this so early when this method was still like a method that not everybody trusted.
Right.
Like
right.
now,
yes,
digital pathology, like not everybody trusts. They want others to do, even though it has been validated so many times already, right?
I mean, and if you've done IHC by hand, maybe in your early days, like it works, some of the time, and then
do it again.
Some of the times it doesn't work, and maybe it's twice as dark on Tuesday as on Thursday, and as we know, like that just doesn't work especially for semi-quantitative companion diagnostic type biomarkers. So that's pretty much me, and I'm as I said, I'm thrilled to be here today with you. chat.
Let's talk about the Roche Digital Pathology DX system. You guys recently received the clearance for primary diagnosis. Can you give us a quick rundown of what the system is all about and how it fits into the digital pathology landscape?
Sure, I'd be happy to. So you mentioned the actual name of the system, and this will become important in a moment. It's called Roche Digital Pathology DX, or we abbreviate it RDPD sometimes. So if you hear me say RDPD during our chat today, you'll know what I'm talking about. So the system, we'll go into this in a bit of detail for the listeners who aren't familiar with this. The system, according to the FDA, has three basic components. So there's a scanner component, a viewer component, a software viewer component, and then there's a display monitor component. And actually in, I believe it's 2016, FDA put out a guidance document that kind of told the world, at least in the United States, how it wanted to look at these systems. So that, and as you mentioned, we had a great year last year, 2 510Ks. In the summer, we had our first clearance, which includes a couple of performance studies, which I'll go into. And then we had a second one, it was called a special 510K. And that is for occurrences where you're basically adding components, but you're not changing the intended use. And you're not really making major changes to it. So we presented an argument to the FDA that the two scanners, the two DP200 and the DP600, were identical by design, even though they were, they were actually manufactured a few years apart. And but they make the same file type, and they have the same so-called pixel pathway. We'll go into that in a little more detail. So and at the time we got our clearance last summer, it was the 5th overall whole slide imaging system clearance with a novel scanner. So I'll explain what that means in a moment. And then again, this is a 510K, so the proper term is clearance and not approval. Just for the
that's like, I think, the most confused term when you talk about something that FDA said yes to, and people confuse.
So it's a cleared system, and generally when you clear something, that means there's is substantially equivalent to a so-called a predicate device. So we'll go into that a little bit with our studies as well. So that was the 5th system. As of today, there are actually 7 now whole slide imaging systems with novel scanners. There are separate clearances for viewers and monitors. We won't talk about those today. And we talk a lot about the slow adoption of digital pathology. I know this was popular in the mid-teens of this century because there were either none or very few cleared systems, and some labs didn't really want to adopt unless the FDA had blessed it accordingly. So the first clearance was 2017. The second one was in 2019. I was actually part of that. And so now we're up to 7. And now it's 8 years after the first clearance. So what I'm getting at is that we can't really use a lack of choice or lack of options as an excuse anymore. Now, there may be other excuses, but we can't use that one as an excuse anymore. So, and then maybe just an understanding in why system. The idea here is that the comparator device for pathologists to make a diagnosis is the microscope, right? What do you need to make that work? Well, before there was electricity, you needed a good mirror lined up against the sun. But now that we have electricity and light bulbs, light sources, you basically need an electric plug, and then you pretty much trust the pathologist, to do what they do. A scanner will not serve that purpose, right? If you have a slide scanner on your desk, you put the slide in, it scans, and then you can't, you need something to read it, right? So that's the rationale for the system, is that the scanner, at least the way we make them today, is not useful as a comparator. So you need to add viewing software. And so our viewing software is called ViewPath. It's now called Navify DP, Navify Digital Pathology. And we had to choose actually a specific monitor, and it was an ASUS brand monitor. So what does the FDA clearance allow us to do? Well, it makes us to make specific claims that are really no different than all of the other whole slide imaging system claims. In fact, if you line up all the intended use statements for all of the systems, they're like identical septuplets. They're pretty much the same. Yeah, the only components are the only difference is rather the different components and the system. names.
Mm-hmm. And I want to highlight this, highlight that you are highlighting that it's a system, because the spoken shortcut is a cleared scanner. But there is no such thing as a cleared scanner, and we keep referencing the pixel pathway. So I'm gonna just let you talk about it whenever you want to touch on this. But that basically defines the system.
So you could talk about the pixel pathway within the scanner. I think what FDA is really referring to is the pixel pathway of the system that
Mm-hmm.
includes the scanner. So what I
Mm-hmm.
will say is that and we'll talk about this a little later on, the algorithms that have been cleared by the FDA for use, there's a very small number of them. But I remember hearing this, but I tried to look it up in a document. I couldn't find it. The position has been that in order to get an algorithm cleared through the FDA or approved, if it turns out to be, it has to be done on a previously cleared or approved system.
Mm-hmm.
So the idea is that it's like you're building a house, right? The foundation might be the scanner. And then on top of that, the walls are the viewing software. And then maybe the second floor is a viewer. And then on top of that, maybe there's some software functionality. That's your crow's nest or your 2-car garage, or however you want to look at it. So the idea is that the system is really the foundation of kind of everything you need to do going forward, including algorithm development or any technical advances.
We recorded a demo video at USCAP. I was at the Roche booth, and you are going to be able to see it on YouTube, the Ventana DP600 scanner. But today I want to dig into your validation study. So the scanners are going to be separate, and I'm going to send everybody who is on my list an email about that as well. But I'm interested in the validation. First, at the very high level, it is logical. FDA wants to know if digital pathology is non-inferior to light microscopy. Like you said, pathologists have been doing their job on the light microscope for a long time, and the microscope is a system in itself. It has like all the components, but because we went digital, we kind of dismantled the component from this one device, but we still want to know that this is non-inferior, because this has been used for pathology since its invention.
Few
But
centuries.
how
Yeah,
do
few
we
centuries.
prove? For centuries. Now, how many
Yeah.
centuries? 2 centuries? 3 centuries? I don't know how many.
Depends on how far you go back. You're going
Right,
back.
depends.
Almost two.
But
Almost two. We'll put it that way.
exactly. But how do you
Yeah.
prove that something like that has been in use for so long
Yeah.
is like the new thing is the same as the old thing, even though they look
Right.
totally different? Like at the high level, what does the FDA want to know?
Sure. And now we have 7 examples that have been published in decision summaries. So I would say the best way to get a sense of how the FDA looks at these is to first look at that performance document in 2016, and then look at the summaries. And then we have a paper in press, as I mentioned, that'll go into a bit more detail. So they're looking at a couple of things. And I will say, you mentioned validate. Like we're a medical device manufacturer. We're not a lab per se. So the V word means different things, slightly different things to medical device manufacturers than to a clinical laboratory. So I'm talking now about the former. So they want an assessment of precision and accuracy, and these seem very intuitive and foundational for really any device, right? But because there are so many cleared systems, they're all equivalent, substantially equivalent. The study designs are fairly fixed. So you have a little bit of leeway there, but not much. And actually ours kind of rolled out at the beginning of the pandemic. And that'll be a really interesting tie-in I'll make near the end. So the goal is that you have these 2 studies. They're fairly sizable. One measures precision, one measures accuracy. And you have to meet so-called acceptance criteria. So you have to decide ahead of time, like what level of precision or accuracy you need to achieve. And then the goal, of course, is using statistics to disprove the null hypothesis. So the null hypothesis in this case would be that digital pathology is nowhere near as good as microscopy. So to disprove that, you need to actually, the way we do it is take a difference in accuracies, and then that difference has to be a certain amount.
So what is the, like, high level, what's the difference between precision and accuracy, and why does the FDA want to see both? Because like you say, device manufacturers have different requirements than if you would take a device and then validate it in a lab, right? There you don't have to do so much, but here you do. Why do you need those two components, and what are they showing?
So I will say, just before I get into that, this is an example where a single person with a single idea, and let's just say they were an octopus, they had lots of hands, they still couldn't do it by themselves. So as a pathologist, I have some idea of what would convince me, but I'm not what I would call a card-carrying statistician. So what I will say is that in the design of these studies, and of course in the analysis of data for anything, for any health authority, statisticians need to be involved so that you have proper study design, and then it's powered adequately enough to rule out any confounding sort of not irrelevant or biased explanation. So that is one reason these studies are pretty juicy. I'll get into that in a moment. So I would say the philosophical question of why is well, let's just say, why don't I go into what we're actually measuring?
I love that you called it philosophical question.
Well, again, some of it's history, and because you're dealing with the substantial equivalence, you can't rewrite the book, right? You've got the predicate, and you have to show your equivalent, and that's pretty. I want to say it's pretty locked in. But let's step back and ask, like, what is primary diagnosis, right? Primary diagnosis is when a pathologist looks at a slide, and through really kind of a miraculous process, the visual information goes in, something happens in the brain, and then, either instantaneously or a few minutes later, or maybe after some tests, some words come out. The words get into the report, and that dictates like everything to follow with the patient. So what's in the black box of the brain that's going on? The way I think of it is that there's kind of three components of primary diagnosis. So one is something called features. Features are what you learn in histology class. Here's a nucleus, here's a cytoplasm, here's a fat cell, here's an infectious agent. The second one is context. So you can probably think of some examples where if I show you some features in this one history, but then I show you the same features in a different history, your primary diagnosis might be different. Right. So it turns out the context can be really, really important. We'll go into that in a little bit. And then the third one is language. So think about, how important that we all have shared meanings of a set of words that come out of the end, right? Invasive versus in situ, pleomorphic versus anaplastic. Like these automatically, whether you like it or not, I say these words, and the part of your brain that makes pictures is already flashing pictures in your head. We don't really understand what's going on. All we have is really, the diagnoses to compare. All right. So, and then of course, my favorite study around making diagnoses is that, like, it's not specific to humans, right? Remember Richard Levinson's famous paper where he trained pigeons to read breast cancer, benign versus malignant.
I know.
With a little bit of, caloric incentive. Nevertheless, like, the pigeon version of Keith could probably do a pretty good job, doing what Keith is trained to do. So that's an amazing insight. It's a very evolutionarily deep process, probably relates to visual cues that help you find food and survive and mate and stuff like that. All right, so the precision study then looks at 23 pre-selected features that, again, they were already baked into the prior predicate devices study, so we didn't really have a choice in those. You would recognize all of them, right? There's things like, adipocytes, skeletal muscle, nerves. There's some abnormal things as well. There's no comparator group, though. But you do have multi-reader and multi-site, multi-scanner and stuff system. So in the end, for that study, we had almost 5,000 instances where a pathologist looks at a region of interest, and they're supposed to identify a feature. So 5,000. So that's like, 4 digits. That usually makes the statisticians quite happy to have an N that high.
Yes.
Yeah, right.
I remember
so then the other study
from my PhD where we had like 3 mice per group. One died, and the statisticians were like, "No way, Yeah, yeah.
Shh, don't tell
The
anybody.
higher
Yeah.
the number.
But really, there's no reproducibility crisis in science, really. No, just kidding. Um, all right, so then the other study, the accuracy study, that's like the granddaddy study. That was, in our
Mm-hmm.
case, 4 sites, 4 pathologists per site, over 2,000 cases. And the basic design is that the pathologist looks at a case with microscopy, and then they look at the same case with like a washout with digital, or vice versa. They're
Mm-hmm.
randomized and
Mm-hmm.
they're put in smaller groups and they're mixed up and so on. So if you add all of that up, it's almost, I think it's 15,000 ish diagnoses are made. So 15,000 instances where a reader looks at a case and says, yes, it's basal cell carcinoma or it's a hernia sac or whatever. And then that wasn't enough actually the endpoint was the disagreement rate. So it's the disagreements, the clinical diagnostic disagreements that make a difference in patient care. And so we train them. So we have, in our case, we had 20 adjudicators. So we train them to judge, like, is this the same as that, or is it a minor disagreement, in which case it's counted as an agree, or is it a major disagreement because it has a change in clinical diagnosis. So when you add that all up, there was like over 40,000 adjudications. So 40,000 instances where an adjudicator, pair of adjudicators actually look at 2 diagnoses blinded, and then they say, these 2 are the same. There's a minor disagreement, which means there's no change in clinical management, or there's a major disagreement, which means there's a change in clinical management.
Mm-hmm.
So these are both types of studies, multi-year. They take thousands of hours of time on our side, hundreds, thousands of hours of reader time. And so these are massive. These are massive studies. And now, again, as I said, there's 7 cleared systems. So they've all been through
So
this
how much
scale
time
of
did it
testing.
take for you to run the study? Because you say you had 4 pathologists per institution. That's like coordinating 16 pathologists. Coordinating
Yeah.
one is a challenge.
Well, right, and there were site leads. There were medical directors at each site that we worked with. There were co-authors on the paper. There's histotechnologists and scanner technologists at each site that are trained on the system. The systems, because they're investigational use only, are not allowed to be tracked onto other side projects. They have to be dedicated to this purpose because they're investigational. So the site has to be on board. The pathologists have to be on board. And they have to be able to dedicate a certain amount of time over weeks to months to get through the workload. In addition to, of course, we all know they have lots of free time, so it's really not a problem.
Like everyone, right? It's a side project, not.
Yeah, exactly. So when you add it all up, it's thousands of hours, and then it's years from the time you think of it to the time you get your protocol written down, your IRBs done, get the machines on site, and then I think for us it was a couple of years, but till after we got the results to get the clearance.
So let's talk about the paper. Your team's paper, which is in press in the American Journal of Clinical Pathology, it mentions specific acceptance criteria for precision and accuracy. Are the numbers that you mentioned, is that mandated by the FDA? Can you walk us through what numbers you need to hit to get FDA clearance, and how Roche system performed against those benchmarks? I'm especially interested in the non-inferiority margin, because you mentioned they are basically quantifying how people disagree. So how much can they disagree? There is an non-inferiority margin of minus 4% that seems to be consistent across different whole slide imaging system throughout the industry. Like, where does it come from? Are we so consistent? Like, what are the numbers that you need to hit in addition to like how many cases you need to do for the statisticians?
Right. So I'll say thank you for both studies, those cutoffs, the study sizes to some extent, the power calculations, they're pretty baked in. You don't have much flexibility. And the goal there is to, I'll explain this when I explain the acceptance. is there to be no doubt. There's no statistical doubt that your data is telling you what you think it is. In other words, you're not achieving acceptance kind of by accident, right? So the studies are sized that way. So that's like impossible from if you talk to a statistician. Okay, so for the precision study, remember I said there's like almost 5,000 instances where a pathologist looks at a screen and says, and looks at a list of features and says check or not. We landed around 90% for all of the metrics, and that included like inter-site, inter-day, inter-reader, and so so the acceptance was 85%, and again it's the lower bounds of the point estimate. So the point estimates were all around 90%. When you added in the confidence intervals, the lower bounds subtracted about 1.5 points. So we were still above the acceptance, which was 85%. And 85% is, to me, it's a common number you encounter with regulators where I think they would, if let's just say the acceptance was 70%, then they would have to go to the public and say, well, 3 times in 10, this test doesn't work, but, 7 times in 10, it does. That doesn't look very good for them, or really for anybody. So 85% is a compromise, I think, between what looks good and what's kind of achievable within the study domain. So we passed that. So we were thrilled to hear about that. All right. So then the accuracy, the metric was the difference in accuracy of each modality. So as I said, they read all the cases or some of the cases manually, washout digital, or they read them digitally, washout manual. So every pathologist reads the case twice. And so if they do have recall bias, it's sort of spread across all the cases. And there's the washout kind of, prevents them from,
I did a similar exercise for a different validation of a project, and I was afraid that I'm gonna very much remember everything I saw. I was surprised how much I have forgotten. Because when we were discussing the study, I'm like, what do you mean it's going to be skewed by the recall bias? And they gave us enough time. What's the time called? Washout period. The washout period was long enough that I actually didn't remember. Or no, in some instances,
that's
I had no idea.
Right. That's the goal, right? There are some pathologists who, self-proclaim that they have memories like elephants and they never forget
not
a case.
Me. I already
and that's probably
myself.
True, right? There are some, so, but if I threw, So, anyways, that is what it is. If there's, and again, your results aren't driven by one reader because there's 16. So if you do have one elephant in the room who's remembering everything, they're only 1/16 of the data. All right, so let's just say the manual accuracy was 99% and the digital accuracy was 97%. So the metric was DR minus MR, which is -2 in that case, okay? So if the acceptance is -4, you had to be greater than -4. So -2 is greater than -4. And actually, I just gave you the point estimates. If you include the confidence intervals for studies of this size, it drops it down about a percent. So where did we land? We landed at -0.6%. With confidence intervals that overlap 0, and the lowest bound of that confidence interval was about 1 point lower, so -1.6, again, which is still, I'll call it miles away from -4. So we were quite happy there that we passed. And so an interesting thing, I think now that all of these studies is, again, there's been 7 clearances, so there've been 7 studies of kind of the scale. 6 out of 7 of those, the difference in accuracy overlaps with 0, and all 7 of 7 actually were greater than -4%. So that, to me, in addition to each system being passing, it tells me that DP is passing, right? Well, I think we'll go into that.
Yeah, that is my impression. It's basically what it is.
Yeah. So that's good news for DP. It's good news for the systems, obviously. And it's a vote of confidence, I think, for the non-inferiority of the two.
Do you think that because of that, there's going to be No, there's not going to be like relaxation of anything because every device is a new device and that's just the norm and people will expect that. I was just hoping like, oh, maybe then people don't have to do 5,000 slides or whatever, 2,000 cases because it actually is good enough.
There've been a few clearances based on viewers that had much smaller cohort size. The risk with a smaller cohort, of course, is that with the confidence intervals, you won't pass, right? So,
That is true.
and we'll go into the factors that contribute. As I said, our study, all the studies, have a matched digital and manual group, and so the endpoint is the difference between the two. So what that means is that whatever environment is contributing to your modality accuracies, should be contributing equally to both of them. And what you're really looking for is a signal that shows that one is actually inferior. And so the fact that they overlap with 0 says you can't find any, which is good. Again, it's a vote for each of the systems. It's a
point,
vote
right?
for digital pathology in general. Yeah, absolutely.
So were there any particular tissue types or any diagnoses that were really good on digital and not so good on the microscope, or the other way around, that they were good, like very concordant between pathologists on the microscope but not so great on digital? Was there anything specific there?
So there are signals, I'll say. There's really nothing specific because none of those subtypes, subgroups, organs are powered appropriately to confidently state that there's, for example, you can't read liver biopsies with DP. None of them had enough sample size to even show that, even if there was a big difference. So we controlled for that 2 ways, and this is similar to where the other groups looked at it. First of all, you're not allowed to front load the cohorts with a bunch of easy cases, a bunch of BCCs and hernia sacs, right? You actually have a predetermined list of organs and diagnoses and the proportions thereof that each site needs to pull out of their archive and enroll. And they do it pretty much consecutively, but they also have targets for specific organs. And we had to meet, most of those. So there were 20 organ groups. And when we do subgroup analysis of the 2,000 cases, 20 groups. And when we do subgroup analyses, the difference in accuracy for every one of those groups was within plus or minus 4%. So again, not powered appropriately, but in other words, there were no organs present that were really driving a good or bad accuracy with one of the other modalities. So the differences were, within a range over all 20 organs. All right, so then we subdivided those 20 into 74 organ plus, diagnoses or procedure types. And these are all predetermined as well based on the predicate device. And so there was a little bit more of a spread. They kind of ranged from like -8 to +8%. So they weren't all squeezed into 4, but it was really interesting looking at the ones that kind of landed at the margin. So it turns out 2 of the ones that were the most accurate digitally, believe it or not manually. And again, underpowered, we don't know, whether it's real. Bladder neoplasia. So why is that? And I start to thinking, again, this is a hypothesis. Well, if you looked at bladder biopsies, like they look like 64 continents on a projection map, right? There's tissue everywhere. They're really challenging to look at by microscopy, and maybe digital makes them easier, right? So maybe you can actually be more accurate. Again, underpowered, all the disclaimers, but I thought that was really interesting. 2 out of the 74 were bladder cancer categories, and that was more accurate digitally. Now, at the other end, more accurate with microscopy were 2 of the breast in situ carcinoma categories, both resections and biopsies. And when I say more accurate with microscopy, again, not much, like 8%. So it's again, if the entire study were 2,000 cases like that, I suppose it would have failed, but we don't know because the study was underpowered with that sample type. And then, but what that sample type tells you is that that's a type that's intrinsically challenging with microscopy, right? atypical ductal hyperplasia versus carcinoma in situ versus microinvasion. So we saw a lot when we actually went back and looked at all the diagnoses, and I'll get through that in a moment. We saw a significant number where, where they were both similar but not exactly right, and the adjudicator called it one way, and sometimes they called it the other. But again, breast in general, this dysplasia carcinoma spectrum, intrinsically challenging with microscopy. So it's not a surprise that it's challenging with digital as well. Let's see. So then when we looked at all these subgroups and we chopped up the data several different ways, we didn't really find any system specific or DP specific pitfalls. So in other words, the statistics told us they were non-inferior, and then when we looked at subgroups, we didn't see a real consistent trend with any of the non-statistically significant subgroups, beyond those that I mentioned. And most of the rest were within this plus or minus 4% range when you looked at difference in accuracy.
So these were organs or types of diagnosis, and in the paper you mentioned specific microscopic features like nuclear grooves that were sometimes challenging to identify digitally, and we're going to explain what these are, especially in the context of Langerhans cells histiocytosis. But then you had perfect identification of the same feature in other contexts. So there's one, and you mentioned that there is different context to the same feature at the beginning, right? So there was one context of a feature where people didn't recognize it digitally, but then there was the same feature, but in a different context, like thyroid cancer, where they did recognize it. So what does this tell us about how pathologists recognize features digitally versus glass? And does this inform in any way the training of pathologists when they transition to digital?
Yeah, those are really good questions. So remember, precision study did not compare to microscopy. So that would have required us having a microscope set up and an arrow saying what's this feature on the microscope, and then doing the same thing digitally. So that was not done. So we can't really say anything about comparison to microscopy with the precision study design. But I think your questions raise a kind of a larger question of, well, why isn't it 100%? Like, can you only see 9 out of 10 features with your system? Like, what's going on? And it turns out that's, that might be the null hypothesis answer. But what happens is actually, if you look at what they chose instead of the right answer, it tells you a lot about why they chose that answer and why they missed it. So I'll explain that quickly. So you mentioned nuclear grooves. Yeah, nuclear grooves were the troublemakers in our the study is designed by having 3 different cases with a feature, and so our curator chose 2 cases that are what I would call textbook: thyroid cancer, ovarian cancer. So these are things like, if you're a medical student, you're learning about these. It's like, oh, nuclear grooves. If you see those, you think thyroid, you think ovarian. And there are a handful of other things you think of as well, but medical students can't memorize everything, right? So there was a case called, you mentioned Langerhans cell histiocytosis, which is a childhood disease, and these are characterized by sort of marginally neoplastic Langerhans cells. They accumulate in the body. There are different forms of it, but they have nuclear grooves, and they're immune cells. So for some reason, only 2 out of 72 instances where the pathologist looked at those LCH cases and called nuclear grooves, they got it right. So only 3%. It was abysmal. And that accounted for nuclear grooves being the worst feature identified in the entire set of 23. So that really, dragged down our results. But when we looked at what they chose instead, it was 2/3 of the ones of the 70, of the 72, the 70, let's say 2/3 of those or more chose so-called Reed-Sternberg cell. So Reed-Sternberg cell, as you know, is the pathognomonic cell, malignant cell of Hodgkin disease, a classic Alzheimer's, right? It's a 2 big nuclei, 2 big nuclei, two big nuclei, and they're kind of next to each other, and it's like the owls hooting at you. So they called, most of them called it that. And then if you look at kind of the background of that lesion, it has a benign infiltrate. These are kind of funky looking cells. A lot of Reed-Sternberg cells, as you know, are what's called non-canonical. They don't, you can find owl's eyes, but not everyone, has those owl's eyes.
So they basically
so
look
we,
like
we can
nuclear
infer
ghosts.
that, yeah, we inferred that they kind of looked at the whole background. They saw these big cells that were, kind of bordering on multinucleated. And they said, Hodgkin's, it must be it's Hodgkin's, must be Reed-Sternberg. Check next case. So that was one reason I think that's plausible based on our data. So then the second reason they missed it was, I would just say, atypical example, right? We all take histology. We learn these beautiful prototypical examples of a feature. The one that we highlight in the paper are called asteroid bodies. These are features of granulomatous and other types of inflammation. And they basically look like big red starfish or octopuses. They have long stringy arms, and they have a nice red center. They don't really look like asteroids. But
whoever is going to be watching
bodies
on this on YouTube, it's going to be on the screen. Don't worry. So if you're just listening and you want to see them, we're going to put them on the screen. No worries.
Excellent. I'd love to hear that. So what we found, so these also, these are features, but they don't occur alone, right? They typically occur inside of a giant cell with kind of a foamy background. And there's some great EM studies that have been done like in the 60s that these are made out of some sort of filaments that nobody still understands. So what we found is that the ones that were really classic canonical looking ones, 100% of the time, they got those. There were others that looked like they were kind of on Foss cuts or maybe deeper cuts and had some of the background, but the features of the asteroid body were really minimal. But they still got those like 80% of the time. So in that case, it tells me that like even though it was a really bad example, they got the context and they said, oh, it must be an asteroid body because that kind of looks like a poor asteroid body, we'll say. So that was the other one. And then the third reason they didn't get 100% was that, once again, let's see, we had 6 readers, 5,000, almost 5,000 instances where a reader is looking at an ROI, looking at a checklist, check or not. And what we found is that there were many, many instances where when they missed it, it wasn't that the feature wasn't there, is that wasn't the feature chosen by the curator. Because the study design only allows the curator to select one feature. So and the most prominent example was osteocytes versus osteoclasts. Okay, so think of those, right? An osteocyte is embedded in bone and it's got arms, but you can't really see it. It's kind of squished by the bone. And the osteoclasts are, bone resorbing cells. They're multinucleated. They don't look anything like each other.
No.
But they often appear
are like
together.
In different places, but you're going to find them in the same slide.
They're in different places, but we don't have an arrow in these ROIs saying, here's your feature. What is it? It's not name the feature. It's find the feature and then name it. Yes.
Okay.
So what we found a lot of the times was that it was a bone section. They picked osteocytes, but it was one where there was an osteoclast in it. And they said, it's not, and they were allowed to select both. But I guess my argument here is like, all right, I'm looking at 1,000 ROIs and I don't want to select. So I attribute that kind of to fatigue, maybe. It's like, this is a repetitive feature hunting task. And like, I have to find 3 of them or 2 of them to get it right. So I think I use that as kind of the third bucket of explanation why people missed it. So in no cases was the feature not evident, or was this, there was a few cases where they said, well, this isn't in focus. I can't see it. But that study design did not allow us to rescan the slides, which is in contrast to the accuracy study, which did allow pathologists to rescan slides if they felt it was a bad scan. So anyways, we have good explanations, I think, for why it wasn't 100% and it had nothing to do with DP.
Yeah, that's kind of a common thread, that it's how people work and it's because it's people, and it's not pathology is an interpretative science. So like you say, it had nothing to do with the method. It had everything to do with the art or science of pathology and the people who are practicing it. And there was another thing that you described in the study, that the longer, more detailed sign-out diagnoses were actually associated with higher disagreement rates for both digital and microscope read. And I don't know, I didn't think this was so intuitive, because I would think, okay, if it's so detailed, like described in such detail, there should be better agreement. But what do you think that reveals about how we would maybe approach diagnostic documentation in pathology reports? Or like, what does it say about what you guys did?
Well, there's certainly a need to make diagnoses more patient-friendly, right? There's a lot of efforts
That's a
undergoing
that's a
in that
topic for a
area.
different
This is
discussion,
not
I
about
guess.
that. It's a topic for a different discussion. In this case, I will say that remember our endpoint was DR minus MR, so the difference in accuracy. Well, in several of the other digital pathology whole slide imaging system studies, a separate primary endpoint was the digital modality accuracy by itself. And I think for reasons that are historical, we did not declare that as an endpoint in our study. Our study kind of took off during the pandemic, and it was very difficult to get feedback during that time, so we had to take some risk going forward. So that endpoint was not declared as an endpoint prospectively, but of course it was measured. And then when we actually put our application in, the FDA said, well, they said, well, what about this endpoint? All right, so it turns out that our modality accuracies were each around 8%. So in the historical acceptance criterion, again with the confidence intervals, lower bounds, etc., the cutoff is 7%. So I will say, if we had declared that or were held to that, we would have failed, and I don't know what would have happened. But we did have to do a pretty in-depth, multi-month root cause analysis to understand, like, why were we landing at 8% with both modalities? As I said, when we subtracted them, they were less than 1%. But viewed separately, they, at least the digital one, would have failed. So we did a root cause analysis, and it turns out we can blame the pandemic. What happened was, when we were in the contracting phase with each of these labs, like, they were all scrambling to get their work done, right? The digital wasn't really, we remember that time, right? Digital was only used kind of spottily. There was some emergency EUAs and enforcement discretions and things. So all the sites we were working with, we worked with three clinical labs and one academic lab. They were all game. They all wanted to participate, but they said, we have to enroll the cases more quickly. So what they ended up doing was not shortening or focusing or, I'll just say curating the diagnosis to kind of what the primary diagnosis was. So we all know pathologists are notorious for hedge language, for verbosity, for use of different types of terms. So we know that, right? So what happened was, for lots of the cases, is that they like, the whole report was just pasted into our database, and then the adjudicators had to sort it out. All right. So then let's give you an example. Okay. So let's just say the reader sees a case and they say, oh, this is diagnosis A. But the reference is A and B. Or maybe it's A, but I can't rule out C. So do you call that an agreement because they both got A? Or do you call it a disagreement because they missed B or C? The adjudicators were, per the designs of these studies, they were given instructions on how to adjudicate, but they weren't overridden. right? Their opinion was their opinion. They looked at this, they looked at that, and they said, is this an agree or disagree? And we trusted them to do that. Now,
Okay.
another unique feature of our study is that we had 20 adjudicators, and they worked in teams. And so when we did our analysis, we ended up seeing kind of a similar phenomenon across all of the adjudicators, and I'll explain that in a moment. So what this teaches us, I think, is that, any kind of absolute modality accuracy in a study as an endpoint is not a good endpoint, right? It's very risky because there are all kinds of other factors that can enter into the study design, execution, data analysis that could get you, let's just say our final number was 20%. Everybody'd say, oh my goodness. But then what if manual was also 20.1%? So the difference is, 0.1%. So I think it's dangerous actually to have modality accuracy by itself without some context as an endpoint in these types of studies, for sure.
Yeah, I see it as like you are assessing something that's already an interpretation by somebody who didn't interpret it. So you, like you say, you add more variables to the system that you're evaluating, like because you already evaluated the features, and the assumption or belief is then, okay, whoever saw the features can take the features and make whatever the diagnosis should be out of those features. And this is where interpretations comes into play, that results in a report that may be convoluted because we're pathologists. So I can totally relate to why this adds another layer of complexity.
A little bit to add to that. Remember, the feature list was pre-specified. Now, some of those features might be in the actual cases. Some might not be. Some may never be, right? The cases in the accuracy study are really designed to be kind of consecutive, a broad representation of what a pathologist in general practice would see with respect to primary diagnosis. And then the last point I left out is that to prove statistically that it was the length of the diagnosis and not difficulty or any of the other 50 variables you could think of. We actually did a pretty strict post hoc statistical analysis. We divided all the input diagnoses into long and short, and it turns out the ones with the longest diagnoses, the half, their disagreement rate was twice what the short diagnoses disagreement rate was, and that was controlling for designation of case difficulty, which is something that it was a type of case that had to be designated input. So we felt like we had really strong data to justify blaming the pandemic. I'll say it that way. And that modality disagreement rate really has, again, nothing to do with the system.
So your data showed that 20x scanning was good enough for almost all diagnoses. So did pathologists actually need 40x? And if they did, how did you use this in the study?
No, that's a really good question. I will say in the precision study, we had a set of features that were kind of high power features. So those were 40, and then shown at 40x. And then we had another set that were lower power features, and those were shown at 20. So it was tested, but we didn't test the same feature at 20 and 40, right? That would be a different study design. So, for example, could you see, salt and pepper chromatin better at 40 than 20? The answer would probably be yes, but we didn't test that. There's kind of high power features and low powered features. And then for primary diagnosis, 40x scans on, almost all the scanners are beautiful, right? The question is whether they're necessary. And if you think back to your pathology training, right, a lot of primary diagnosis is 1x or 2x, right? It has a certain appearance, and then you really just need to go down to 20 or 40 or even sometimes on oil to verify something that is a lower power impression. So, and then consider that the 40x, like a lot of the scanners in this class can do 20 and 40x, but by definition, without compression, the 40x images are 4 times bigger, and they probably take 4 times longer to collect, roughly. So the question is, do you
Yeah.
need that for primary diagnosis? And then the final piece of it is that our predicate device tested 20x. So we were kind of on the hook to, to test 20x. If we had tested 40x, it would have been a different study. So our scanner, like the predicate device, will do 20x and 40x scans. So anyways, lots of background to that. But the bottom line is we scanned everything at 20 and then gave the pathologist the option to scan at 40 if they felt like, well, this isn't good enough, or I need to see a higher power image. And so it turns out, let's see, there's only like 3,200 slides in the study, only 6 slides out of 3,200 where the pathologist said, I can't see this at 20x, I need to see it higher magnification.
Oh, really?
So that's far less than 1%. And I think, again, that's kind of what the predicate device study showed. And it also says that, 20x is fine for the vast, vast majority of diagnoses that happen in general surgical pathology.
So was there anything specific? Was there the pattern? I know it was only a couple of slides, but like, what did they ask for? Why did they ask for 40x?
Yeah, and we didn't. Well, I'll just say that we have that, but the number. The number was so small that you couldn't really, like, if I told you 2 of them said that I couldn't see the chromatin, like that, you could state that, but that's like far less than 0.1%. So you can't really make any conclusions about that from a statistical standpoint. But on the other hand, like, people have looked at this in the digital pathology literature, and then also some of it is intuitive, right? We know that there are some features that you need to kind of focus up and down to get really confident about. So, and that made sense with the cases that we saw that were challenging in both modalities. So like fine chromatin features, dysplasia versus atypical nuclei, things like that. Looking for H. pylori. But interestingly, once again, 20x, all the rule out H. pylori cases, the difference between the error rate, or the major discrepancy rate, again, DR minus MR, was almost 0 for those cases. So even at 20x, there was no signal there that even, looking for H. pylori and gastric biopsies was, more of a problem with 20x versus 40x. So, yeah, so it's now I will say, and I don't know if we're going to talk about this, but, a lot of the algorithms now are trained at 40x or even, different magnification. So 20x is adequate for primary diagnosis, but the question is, will it be adequate for all the algorithms that get made? If they're trained on 40x, that's an open question.
Yeah, then you probably would have to train at a certain magnification. And I'm also thinking, okay, then how can you leverage the fact for efficiency, for like reducing the size of images, the fact that you just mentioned that most of the diagnoses are a low power diagnosis, or are low power diagnosis with high power confirmation. Like you look at low power and you see the focus and you just go to the focus of whatever you're interested in. Like you don't need the whole image at 40x. How can we, and that's beyond the scope of our discussion, but basically to me it's like, okay, it's natural that pathologists don't look at every single pixel of the image.
No, they can't.
Only need a confirmation of a suspicion that takes a lot less than scanning this whole thing at 40x. So that's, food for thought, probably for the technical experts in the digital pathology team. How can you leverage this diagnostic or pattern recognition behavior in designing algorithms, in designing infrastructure? However, right? I don't have the answer to this, but to me it's like waste of resources to do 40x.
Yeah, some context maybe is that, like, when you collect the data in a form called a data pyramid, right? It's analogous to being able to zoom in and out, and you're right. Like, it would be very challenging to look at every pixel of a 40x image of a large resection. Like, that's not how pathology's done. On the other hand, the actual areas where you need high versus lower magnification is very case specific and it's dependent on the pathologist's preference.
Mm-hmm.
So I think unless you get into something like virtual microscopy where you're dynamically judging how much information you have to take in different areas, the way these scanners work right now is that they make a one digital image, and it can be zoomed
Yeah.
up and down. And
Mm-hmm.
then, of course, depending on the lab use case, right, you need a certain bandwidth to manage that data. There's a so-called hot, warm, cold storage that might be useful for initial
Mm-hmm.
diagnosis versus archiving versus secondary consultations. So I think all this
And
tells us is that for
we don't
most
need to have
diagnoses, you
the
don't
answer,
need.
but
Yeah, I'm just saying those are the variables that people are thinking about now.
so looking at the FDA, all of the FDA cleared whole slide imaging systems, they're showing very similar performance in the validation studies. And I kind of alluded to this, but in your paper, you mentioned that these studies, these designs were originally negotiated between FDA and the Digital Pathology Association and various manufacturers. So given that 6 and now 7 different whole slide imaging system with distinct types of scanners have now been cleared with comparable results, do you
Yep.
think it might be time to revise those agreements? And, we already started talking about it, but maybe focus more on image quality and usability testing rather than repeating those like highly resource intensive clinical studies that give us similar outcomes.
Yeah.
What are
No,
your thoughts on that?
you're singing my song for sure. I think I was with one of the manufacturers when we did the performance study for the second system that was cleared, and we were really excited to see that. And this was like 2018, 2019. We were really excited to see that the numbers came out almost identical to the first system. They were like right on top of them.
Mm-hmm.
And so, and I've given several talks on this, and now contributed to the literature in various ways. And in some of the informal talks, I kind of joke. I like to joke about things sometimes. That now with 5 of these, 6 of these, and the 7th one, it's almost like we're rediscovering a constant of nature. We're rediscovering pi.
Mm-hmm.
We're rediscovering the gravitational constant. Like we're seeing the same thing over and over again with very different systems from a design standpoint.
Mm-hmm.
So I think that does raise the question of whether you need, studies of this magnitude after you have so much experience now, almost a decade with these devices. So this is a point in our paper, and for this purpose, we compare our results with everybody else's.
Mm-hmm.
And so these studies are a lot of work. They're quite burdensome, millions of dollars, years. I don't even want to know how many hours they are of people's time. And we saw the effect of basically taking shortcuts during the pandemic when people's time was
Mm-hmm.
was critical. So it does really, I think, force the field to raise the question, regulators as well. Is there a less burdensome approach? And I think the things you mentioned, the system usability, the critical task, the risk analysis around that, is that adequate for primary diagnosis? We can test that today. And then image quality, right? There are actually make metrics within the FDA about what constitutes image equivalence for pixel identical images, and that could certainly be
Okay.
expanded to include pixel non-identical images or images of a particular type. There are metrics to do that. So I think we're entering an era that maybe relieves some of the burden around the creation and definition and updating of these systems in the future.
Mm-hmm. I hope so. I also I kind of secretly hope that after the pandemic, where we had the discretion to use the systems, that this data is gonna support the use of whichever technically good system for generating these images. And I still secretly hope that all this data from all the systems that were allowed to be used then is going to be gathered together with what the manufacturers did for clearances, and that you can have like a giant data pool saying, hey, it's good enough. Like, ask the pathologist if it's good enough for them and let them work with it. Obviously, within a framework, within parameters, and things like that. But I think, like you say, like, do we need to keep rediscovering nature? It's a microscope in
Well,
a box in a different form,
yeah,
right?
how many significant digits, right? How many significant digits do we
Mm-hmm.
need pi to, I guess, at this point? You know, this is why, again, almost 10 years has passed since the first clearance. So
Mm-hmm.
it's always a good time to reassess uh, contemporary
We've been
standards, I would say.
touching and mentioning the pixel pathway. So basically, when digital pathology first emerged, the pixel pathway, the path that pixels take from scanner to viewer to display, was typically maintained within systems from a single manufacturer, right? And now we're seeing systems cleared with components from different vendors. And you guys have your own system, but I know that on the back end of those systems, you can attach algorithms. Can you tell me how Roche is approaching interoperability and third-party algorithms? What is needed in such case to maintain confidence in validated solutions? Because, if you have something from one vendor, they took care of it, but what if you want different components from different vendors? Can you still trust it? How do you guys think about it? What do you personally also think about it?
Yeah, the short answer is it depends on, right, the combination and
It
depends on the
depends.
use. And why do I say that? So the initial clearances were one scanner, one software, one monitor, boom. And
Mm-hmm.
then what now we have with 7 clearances, at least with novel scanners, is really exciting, right? We've got some with multiple file types, or at least additional file types, different like browsers and viewers. Um, and we have like different monitors. There's one of the manufacturers that has something called a PCCP, predetermined change control plan. So this is sort of a regulatory tool that's
Oh, wow.
emerging that can be useful to update your system, in a predetermined, negotiated fashion with the FDA in
Mm-hmm.
this case. Um, and if you're basically making potentially a major change that doesn't really change the intended use, and you do the proper risk analysis and you do some testing, some bench testing that is agreed upon, and you pass that, then you don't need to go through a whole new 510K or even a supplement. You just do the testing, you update the system, and it's a note to file. So this is a potentially less burdensome way to kind of keep up to date some of these systems that are, some of the components are going to be obsolete in a year because the other manufacturers
Mm-hmm.
just don't make it
Yeah.
anymore. And you don't want to have to completely do, another 2,000 case clinical study just to keep a 510K in place. I mean, you don't want to. If you have to, you have to. But that's really not a good use of time, assuming you've done the
But if you
right,
don't have to, then why?
design work and analytical testing. Yeah, but you did mention image analysis. So I will say that that a lot of what pathologists do, for primary diagnosis is grade and estimate quantities, and there's a
Mm-hmm.
huge amount of excitement and activity making algorithms to do some of this. And I just want to emphasize that in none of these 7 studies were primary diagnosis were algorithms used really for any purpose. They're all just about primary diagnosis and diagnostic comparisons
Mm-hmm.
and
Mm-hmm.
feature identification. So, and then if you think about like pathologists, like, you're a pathologist, I'm a pathologist. Some pathologists are blindingly fast with microscopes. And so if you give
Mm-hmm.
them, a clunky user interface and a bunch of screens and a bunch of pull down menus, they're not happy. So
Yeah. Yeah.
and this is the way I think that, all the systems are getting better, but there's a big ergonomic challenge among all of us vendors to really make our interfaces as blindingly fast as the microscope can be. So there's lots of ways to do that. Some of
Mm-hmm.
that's software, some of that's roller balls and extra things like that. That's going to happen. So the point I'm trying to get at now, pathologists can use these systems for primary diagnosis, confidently, but there really isn't a great number of choices for algorithms. Most of the algorithms, the vast majority that are used in the United States are research use only, which means The quality is variable, and the lab's pretty much on their own to validate. So that engenders kind of a huge risk for the labs as to which algorithm to pick and how do they validate it. We as a manufacturer, if we have a research use only algorithm, we can't tell them how to do that. It's kind of on them. So it does put a burden on us, however, to make a good product. We hope that no manufacturers want to put out, a quick product that's not fundamentally good. But so then the question is, how is Roche looking at this? And you may have heard, I work with the commercial team, but I'm not the commercial guy. I am excited about this, though. We do have a suite of algorithms right now, again, all research use only, not part of our primary diagnosis clearance in the United States, that pretty much read our IHC or ISH tests. So HER2, PD-L1, ERPR, Ki67. So these are tools that are designed for things that happen in case level sign out. And so we're interested in taking this through the FDA clearance process, as are a lot of manufacturers. Unfortunately, as I said, right now there's only two predicate device algorithms that are cleared in the United States on FFP sections for use in these systems. And that's, of course, Page Prostate and then IBEX Galen second read, it's called. So this is exciting, but it's a tiny, tiny number of predicate devices. So the way Roche is looking at this is that we've created something called the open environment, which is what I would call a full integration of third-party algorithms capability into the Navify DP environment. So from a case level standpoint, and again, these are all for research use only, one doesn't have to flip between screens or browsers or interfaces. The integration is complete. So when you're dealing with a set of slides, all of the functionality is available to you within that environment. So we have now, I think, 10 plus vendors signed up. I think over 20 algorithms. The first two, this is an experiment. It's a work in progress. I've had good feedback so far. The first two that were fully integrated were IBEX and PathAI. So those are two reputable companies. And we hope to be, the marketplace of choice in the future for this reason. But again, just want to make it super clear. None of these algorithms are part of primary diagnosis. They're research use only. They don't fall under our clearance. And so we just need to be very, very clear about our messaging to customers about, what is FDA cleared and what's not. The second thing, I guess, the point you mentioned, I consider DP at kind of his time in history, DP and algorithms as a bolt-on, right? We've got 40 years plus of using, we're getting better at it, right? Automated IHC staining. We have several predictive companion diagnostics there where pathologists, read the slides and a treatment decision's made. We've got hundreds and hundreds of sort of called class 1 stains in the United States that are used as a basis of diagnosis and prognosis. And everything happens with that being read on a microscope, just fine in the lab now. And now we have all this excitement around big data, digitization. And so we're scanning these slides and we're creating a ton of data. So it's a bolt-on, right? I don't want to say it's an afterthought, but it is kind of an afterthought. We're trying to add it. But then, of course, the question is for any particular AI or really anytime you make the system more complex by adding something, are you magnifying error or are you compensating for it? And I would say we don't know because it's a bolt-on. There's only one way to find out. You've got to do the experiment. Are you getting a number that compensates, for example, for a stain type, pre-analytical variation, or are you getting a number that's actually making it worse, right? Are you worse than the pathologist? So this is a whole area of investigation that needs to happen. I would say the Roche solution there is linking the design of the assay with the scanning and the algorithm by design. Because what that does then is, in my mind, it de-risks the lab's decision on which assay to choose, which scanning system, which algorithm, right? You may pick, this scanning system, or rather this stain, run on this stainer, you add on this scanner, you file it in this format, you run this algorithm. It might work, like it might work fine, might not. You don't know. And I think there are going to be some labs that maybe they like that level of uncertainty because they want to tinker and see what's going to happen. But there's going to be a lot who just don't want to repeat others' mistakes, I think. So I think one of the Roche strengths is integration by design.
Yeah. And I think not only because, there's even more variability because or not only variability, but variables that you add to the system that you have to test, you're testing something fundamentally different than what we were just talking about. You're not testing the pathologist's ability to recognize something and diagnose disease. You're testing,
Right.
and to me, this is like like a flaw of comparing quantitative scoring to a visual semi-quantitative assessment, because how are you checking if those algorithms are good enough? You're checking against your ground truth, and unfortunately your ground truth generator is a human that is worse at the
Yep.
task of quantification than the computer that you're comparing them to. So that's like, to me, that's another whole different way of evaluating this. And me personally, I think it's not being done correctly. It's not being done according to our physiological and visual strengths, but we don't have a better way to do it. And the way we do it right now visually is the standard of care. So then on the other hand, you're comparing to standard of care. So that's
Right.
like a whole other mix up, but I totally um, get the message that you're trying to convey, doing it by design. um, and the next question is, okay, what is the design to then test it and check it and make sure that it's not a lot of uncertainty coming out on the other end?
Well, I do think you bring up a really important point about the pathologist being the ground truth today. And I think if you set up a study for registration or scientific credibility, if you
Mm-hmm.
don't consult a pathologist or use their opinion, it's viewed as like not credible, and you're just you're doing random computer vision. I don't think that's completely true, but I think on the other hand, there will be a point maybe in the future when the algorithms get so good compared to the pathologists that it'll no longer be acceptable to consider the pathologist the ground truth.
Mm-hmm.
The image, computer vision on that's properly tested and will be the ground truth. I
Mm-hmm.
think that's coming. I think we're still a ways away from that.
Yes.
But of course, maybe it's like like Wobegon Days. Do you know this NPR show where like
No,
everybody's above
I
average
don't.
kind of thing? Every pathologist thinks they're above average at. Yeah, no, it's an American radio show. I don't think they do it anymore. But the joke was, yeah, all the children are above average.
Yeah, I guess it's, everybody thinks that they're better than the average. I mean, I like to think about myself that I'm better than the average.
Well,
Um,
of course you are. I mean, so am I.
right?
I mean, right, of course.
Oh, thank you, thank you. But
Yeah.
depends in what. But I agree with what you say. Or there's going to be another like orthogonal modality to check against. And what do I mean by that? So in the first chameleon challenge,
Yeah.
um, that also counts for annotations, for everything that goes into this algorithm development. For the chameleon challenge, where they were looking with image analysis for epithelial cells in lymph nodes, basically cancer metastasis in lymph nodes. They
Yep.
just did IHC with
Yes.
cytokeratin, and that was their ground truth, and not a pathologist annotating that could like miss a cell in the corner of the lymph node or whatever, right? So I thought that was a good way of solving it. Obviously, resource money intensive, but it is an objective method you can compare to. And you know, maybe we need these type of objective methods, and some of them are not developed yet, right?
Well, there's this whole area now of virtual staining that may be with AI that may be
True.
useful for
Yeah,
defining the ground
there,
truth.
there's.
That's
I
emerging.
hope.
Yep. Yep.
I hope stuff is gonna emerge. So, based on everything you've learned from this validation process, what advice would you give to pathology labs that are thinking about going digital? And as someone with your finger on the pulse, and you know, with your experience also, you've been doing this for over a decade. What do you see coming down the pike in the next few years? We can include the virtual staining in this one.
Yeah. No, I'm watching this carefully. I would say my pulse is quickening. It's a really exciting time in history to be, part of this field. I'll start with the second question.
Hmm.
I would say there's still a growing gap between the conceptual discoveries that are occurring with a variety of technologies, including digital pathology, computer vision, and what I see being quickly adapted to actually improving human health. So I'm really focused on that personally for, I think, about the next decade of my life. Like, what does that transition look like? How do you take something that comes out of a Nature paper or something
Hmm.
that and actually benefit patients? I think we still that's hard. That's hard. It's not just science; it's business. It's aligned incentives, multi stakeholders, etc. So, as you know, we have a large amount of information that's a large amount of knowledge in deep learning and AI and different ways to do AI just on H&E stain sections. That's going to have tremendous impact down the road. Maybe not immediately as replacing the pathologist, for example, but maybe being a screen. So understanding what screening performance metrics need to be for clinical use. There's a huge amount of activity you've seen just in the last year or two on large language models, Chat
Hmm.
GPT, agentic AI, and things like that. And this is why I think actually the final output of primary diagnosis, which is all those words, those are all training fodder for these models. So that's really exciting. A kind of a parallel area, as you know, we've followed this area together in the past, is this emerging field called single cell and spatial multi-omic biology. So this isn't
Yes.
really at the initial stages. It's not AI, but it's just generating massive amounts of data on a single cell level, either in soluble situations or in tissue. It's basically a multi-marker analysis. So instead of 2 or 3 markers, on a cell, you can get 1,000 or a whole transcriptome. I mean, it's incredible. These techniques have really just emerged in the last couple of years at this scale. So the amount of data, the amount of discovery that will, I hope, feed into diagnostics is going to be massive. And probably the leading indication would be some sort of use in immuno-oncology, understanding why some
Hmm.
patients respond to
Yes.
checkpoint inhibitors and some don't. It really requires that you get a handle on all the different cell types in the biopsy, not just the tumor cells, which ones are helping the immune system, eliminate the tumor, which ones are preventing it, what is their mechanism. Like, spatial biology tells you that. It's
Hmm.
absolutely amazing. But the question is priority. How do you convert that into actually actionable clinical tests? Go ahead. You're going to say something?
Yes, that immune oncology is where you cannot really go too much further without image analysis. So that is going to be the discipline where all these uh, imaging single cell markers, multi markers um, methods are going to be developing in parallel with the software, or like the image analysis analytics for that, because that's beyond visual capacity of a human. You can confirm something, but there is no way you can analyze it in a meaningful way.
Absolutely. Yeah, it can't be done with the human eye. You can't do it with a place map,
Hmm.
looking with relative abundances. You need computational computer vision, right? Computer vision,
Hmm.
in essence.
Hmm.
And so that, yeah, and yeah, no, that's a really exciting area. All right, so then the first part, let's see. Oh, advice. Let's see. I think I have 4
Yes.
kind of general pieces of advice for labs considering digital pathology. I think, as I said, there's been a slow level of adoption. I think it's still slow, but it's increasing in the past few years, which is exciting. First of all, and again, there's lots of organizations that help out with this. First, I would say, try to succeed on a small scale before you maybe inadvertently fail on a grand scale, right? You have to build a lot of stakeholders to consensus to get the investment and the business cases around digital pathology adoption. And I think most of the labs I've seen that do this, they focus on a few things that are important that help them learn lessons and collaborate with others in the field, other vendors, and others, just other people. The second one, I think, is you need a champion, a local champion, and ideally that person is a pathologist. Somebody, I'll just say coming out of training at the early stage in their career. I haven't met many trainees who are finishing up their fellowships and are champing at the bit to only use a microscope the rest of their life. In fact, none of them are, right? They all want to use digital in the future.
No.
So I would say you get a fellowship trained new partner in your firm or a new pathologist, and you make part of their job being the champion to work with the different teams and help enable some sort of installation that actually will work. Let's see, the third piece is networking. I think, as I said, I've been doing digital pathology one way or another since I entered pharma, 15, 16 years. And I've just been incredibly impressed by how generous people in this field are with their time, how much they want to help people learn. I saw that when I started, I was using, early on, I was using the Visiopharm, early versions of that. And that, they were incredibly helpful to answer the pharma questions we had. Now there's a lot more vendors. There's a lot more Visiopharm still here, but there's a lot more other vendors. There's a huge there are evangelists, I'll call them, in the digital pathology space, the Dr. Parwanis of the world. We need them because they give us hope that almost anything is possible. But there's a huge network of folks that have been trained behind them and that are just as equally knowledgeable in their area. And I found, for example, the API to be very useful for that as an organization. DPA is another one that founded really to nurture this field from the side of the vendor, from the side of the research user, from the side of pharma, now from the side of the AI companies. So anyways, don't be a wallflower, right? Reach out. You don't get what you don't ask for. Ask the question and ask a lot of people. And I guess the last thing I see is the major barrier is some sort of lobbying we need to do, right? It was the American Reinvestment and Recovery Act of 2009 that, at least in the U.S., mandated the use of electronic medical records to get reimbursed. And so that, and then a few years later, as everybody was using, for better or for worse, electronic medical records, we now have, 43 Category 3 CPT codes in the United States. You get reimbursed a total of $0 for those today. So they're experimental.
I know.
It's not quite an incentive yet, but it's a first step. And we still don't have what the professional component of using an algorithm is. What's that worth? I think we're assuming it's worth nothing because it's just the pathologist's job anyways, but that may not be the right way to go. So those are the 4 pieces of advice. Succeed small. Need a local champion, ideally a pathologist. Network like crazy. And I think we need to lobby as a group. And I think there are groups that are doing that, including CAP and others in the United States.
I think
maybe just in the last, just a reflection, if that's all right. Yeah. So, I think it's almost been 50 years since I took my first biology course in high school. So I think about learning about DNA and then, eventually becoming a biologist, a physician, a pathologist. I'm still just in awe at how much has changed between, then and now, last century to this century. And I think, for example, of my grandmother, she was born, no, planes didn't exist. And by the time, she was an older person, we were flying everywhere. So that sort of radical transformation in pathology and how science becomes new pathology, I've seen that. And I think you've seen a fraction of that as well. And now, of course, we have sequencing, we have big data, we have AI, we have all kinds of ways to analyze the data. I'm really excited to see what happens in the next, 10 to 15 years. But I do believe the importance of digital pathology is that I think all of this information, if it is interpreted in the context of tissue, it all converges on the digital pathology work environment. So that's one reason I'm really excited to see what happens in the space, because once again, you can publish a paper in a very reputable journal and show a Tisney plot or pseudotime analyses and all of these really beautiful data visualization effects that are tools that are not pathology, right? Everything happens that is happening in the lesion on the slide that we visualize. So how do we convert that knowledge into thinking about what's actually happening in the disease? And that's digital pathology to me. And that's super exciting.
Thank you so much. So your advice for new labs, I can take it as life advice as well. And like you say, I mean, even me, even though I have not been practicing for 50 years yet, I have seen so many things happen that I'm no longer like thinking, oh, it's not possible. I'm like, let's wait half a year and see what smart people in this field come up with. And now I'm obviously fascinated by the large language models. How can this help us? And multimodality, not only multimodality, the one that we talked about with multiple markers, but multiple data points. And when I was starting, it was like, oh, it's not going to happen in my lifetime. And now I'm like, let me wait half a year and see. So thank you so much for the insights.
No, my pleasure. Thank you. No, it's a pleasure to talk to you. I'm so glad you're doing this for the field. And it's really great to see you.
I hope we see each other at the next conference. And I wish you a fantastic rest of your day.